Next, the molecular cloning lab. This one is a bit more difficult, okay? So make sure you give yourself plenty of time, 2 to 3 hours per simulation, and do them on different days, and really help yourself out with this. Okay, so I went through the simulation, and I had to keep going through my notes and my screenshots to try to piece together, because it really hops around a lot, and there are some areas that can be very confusing in this. I wish there was a different version of this that I could provide to you, because it is a little confusing. So really make sure you reference these slides before and after you do the simulation, so that you can make sure you really understand the flow, what's doing what, etc. Okay, so in the molecular cloning Labster, we will be working with yeast, and we want to be able to determine if a particular gene called the RAD52 gene, if it's important for DNA repair. So it's suspected that it's important, but we want to test that. And in order to test if the RAD52 is involved in DNA repair, you need to be able to expose cells to UV radiation and cause DNA damage. And then after that, you need some way to be able to tell if the cells are expressing this RAD52 or not. So how can you tell if cells that are alive are currently expressing this RAD52 gene without killing them? So the idea here is to use what we call a reporter gene. So here at the bottom, it's typed reporter gene called GFP, and you're probably already familiar with GFP, but it's for a green fluorescent protein. And we actually have gotten this gene from jellyfish that naturally make this green light fluorescence. Excuse me. Okay, so using this GFP gene as a reporter, if we link it with the RAD52 gene such that every time RAD52 is expressed, it automatically expresses the GFP, then we can visually see, we will know if we're seeing GFP, if we're seeing the green light, then RAD52 must also have been expressed, even though we cannot directly see the RAD52. Okay, so that's the concept of a reporter gene, using GFP as an indicator or a reporter of expression of RAD52. Okay, and so this whole lab is focused on how can you make this RAD52 GFP expression vector. And so you need to get the RAD52 gene from the yeast, purify it. You need to get the GFP, and it's actually enhanced GFP to be even brighter. And we need to get that from a vector called a library vector. And I think that's up here. So use a library plasmid to obtain the eGFP gene. Okay, so you're going to do 2 separate methods because you're talking about 2 different genes. So for the DNA extraction from the yeast to get the RAD52 gene, you'll have a whole set of protocols for that. And then to get the GFP gene from the library plasmid, you'll have a different set, a different protocol for that. And then after you've done both of those, then you're going to put those 2 genes together. You're going to ligase or put together, seal together, covalently bond these 2 genes together. So they essentially will function as one because they have one promoter. So the RNA polymerase for transcription will just automatically go down and transcribe both of them. Okay. And together, the RAD52 and the GFP are called the gene of interest, the gene of interest fragment, and then sometimes it's referred to as the insert. Okay, so the insert is the RAD52 plus the eGFP that you've linked together. And you're going to insert that into what we call an expression vector. So up here, we talked about a library plasmid. This is a plasmid that is good with replication, but it doesn't have what it needs to be expressed. It's not, transcription's not going to occur. Okay, so the use of this is that common genes, useful genes that you would use in different experiments are already there on hand, and you can utilize them. So that's a library plasmid. It's just a storage, it's a storage unit of a bunch of different common genes that are useful. And so we're going to use that to get the GFP. Then we put the RAD52 and the GFP together, and that's our insert. We insert that into an expression vector. Expression vector is a type of plasmid that can actually be expressed, made into protein, right? So the genes on the expression vector will actually be transcribed and then translated. So you're going to learn how to insert the gene of interest fragment into the expression vector. And then you finally have your complete expression vector that you can use to transform some yeast. And in this case, yeast are not as delicate as our human tissue culture was. So we will do transformation using electroporation. So you're going to zap these guys, right, with a high voltage, very quick high voltage to cause that membrane to temporarily loosen up and allow that vector to get in. And then lastly, you're going to test if the transformed cells do or do not express the RAD52 after exposure. So see what's the expression look like before and what's the expression look like after you expose it to UV light. And of course, as I already mentioned, using the GFP as the reporter gene to indicate RAD52 expression, you simply just look for green light from the cells. Okay, so now this was the full overview, and I'll go into some more detail of the steps, especially because that will include the experimental overview. So you're given this flow chart, but it doesn't include everything. Okay, so if I were you, if you have a tablet that you can just write on it, otherwise I would either sketch this out or print it out, and I would add in a few things along the way. Because if you only look at this, you're going to miss a couple of key steps. Okay, but in general, this is what we're doing. We have to do a DNA extraction on yeast to get the RAD52 gene. What they don't show is the using the nanodrop to make sure you actually got DNA from your extraction, and then performing PCR to amplify just the gene that you want. And whenever you do PCR, you follow that up with a gel. So here they show gel electrophoresis, but they didn't show it here. They also didn't show the nanodrop. Okay, so this is what I'm talking about by adding in some details through this flowchart. Okay, separately, you will do the GFP extraction where you cut it out of the plasmid, and we'll talk about those steps. And then eventually you're able to put the two genes together with your expression plasmid, and then you will transform some yeast with that using electroporation. And then you'll select for cells that are actually have taken up the plasmid using antibiotic selection. Then you'll cultivate the ones that are indeed expressing or have successfully taken up the plasmid. And then once you have a whole bunch of yeast that have the expression plasmid, then you can finally do your actual experiment of testing if the UV radiation will turn on the RAD52 expression. Okay, so you can refer back to this anytime you want, and just like I said, make some notes along the way. Okay, so the details that you need to know for the RAD52 DNA extraction. So this gene's already in yeast, but we want to manipulate it so that we can see when it is being expressed and when it is not being expressed. So to do this extraction, you will not actually do the extraction, the simulation will do it for you. And they tell you that it's going to be a phenyl chloroform extraction. Both of those are harmful. That would be done in a hood with all the proper PPE. But basically what's going to happen is that's going to lyse open the yeast, and then it's going to make phases, and you'll get one of the phases will contain DNA. Then you will clean that up, purify it, and then you have to ask yourself, is this stuff that I extracted and purified, how do I know if it's actually DNA or if I accidentally took some other part of the cell? We can't see that with our eyes, right? So what we do is we use a nanodrop, which is a highly specialized spectrophotometer that can work with very small quantities. So this is the nanodrops used for all this molecular work. So half up to 2 microliters, right? So super, super tiny volume. And you set it to the wavelength of DNA. So it's about 255 nanometers. You're going to see a range where it shows you, like, I think starting about 230 and then up to like 280. And you'll see the peak is at around 255. And that tells you that that's the wavelength where you get the most absorption of the molecule that you put in the nanodrop. And we know, again, that DNA, the wavelength of DNA should be around 250. And so that makes sense. It means that the stuff you extracted really is DNA if you get a peak at the same wavelength as DNA, right? But it doesn't tell you anything else. It doesn't tell you what the DNA is. It just tells you how much you have, the concentration, and at what wavelength it absorbs, okay? So then we still need to do some work to get just the RAD52 gene. Instead of having all of the DNA from the yeast cell, we want just the gene of interest. So we can perform PCR, and that's going to benefit us in a couple ways. It's going to give us just the gene that we want by selecting the correct primers. The polymerase will only copy that particular region that we select. And it also amplifies it. So it's going to give us a much larger volume to work with, which is really important because molecules are super tiny and very difficult to work with. So we need a lot of it. Okay, so after the PCR, we have to run gel electrophoresis to actually get all of the RAD52 gene that we just amplified into one spot so that we can work with it. So you're going to do gel electrophoresis. Remember, gels separate molecules by size and charge. And so all the similar size and charged molecules are going to end up in the same spots. And that leads to formation of bands. And so the RAD52 gene is going to accumulate in one particular spot on the gel, forming a band, a visible dark spot. Okay. And so then you're going to identify the band that has the gene, and then you'll cut that band out of the gel and do a gel extraction to get your gene out of the gel so that you can work with it. So then you have your purified, amplified RAD52 gene, and you just let that sit in a tube while you do the next part, which is getting your GFP molecule. When you get your GFP molecule, restriction enzymes will be used to cut the GFP out of the plasmid, the library plasmid. And so this is where you'll see an image called a map of the library plasmid, and it'll show you how they made this plasmid to have a lot of different restriction enzyme cut sites, which is a specific sequence. And so you can use this plasmid in a lot of different ways, depending on which restriction enzymes you pick. So the simulation is going to walk you through how to select the correct restriction enzymes. And this part might not be as clear in the simulation, so I want to clarify it for you here. You're going to use the library plasmid map to find a restriction enzyme right before the eGFP gene. And you'll see, and I should have pulled up that image to show you, but there's going to be several restriction enzymes in a row. And some of them in parentheses will have numbers like 1, 2, 3, 4, and 5, right? You want to find which restriction enzyme has just a 1, right? So that sequence only occurs once before the GFP. We only want to make one cut before and one cut after. So then you'll repeat that same method looking immediately after the GFP gene for a restriction enzyme site that only occurs one time. Okay. I can pull that up and show you what that looks like so that you can know what I'm talking about. Okay, so if I go to micro, online labs, viral gene therapy, molecular cloning lab, and I'm going to here. This, maybe I passed it. Just 1 second. I will have the image. Let's see. So it's not this one. I apologize, just 1 second. I just want you to be able to see this map of the plasmid so I can explain these sites to you. Here we go. So let me go ahead and make that full screen. Okay, so this is the library vector map that we're using to cut out the GFP. And the GFP gene is right here. This really bright green arrow represents the space where the sequence for this gene lies. So the arrow represents the gene, and you'll see here the name of the gene is the eGFP gene. And then next to it, it tells you the base pair numbers that it's found. So on this plasmid, you'll find the EFP gene starting at 2506 and ending at 3225 base pair number. Okay. Now, the instructions to figure out which restriction enzymes you're going to use is looking right before and right after, immediately after. So you want to look immediately after the gene and immediately before the gene right here. And notice it shows you where it's located, which base pair number, and then it tells you the name of the restriction enzyme. So this is EcoR1, KPN1, NCO1, XBA1. And it tells you how many times that sequence occurs. So for EcoR1, it occurs 3 times. This one occurs twice. This one also 3 times. And then finally, you see this XBA1, and it only occurs 1 time. So you would want to select that gene. I'm sorry, you would want to select that restriction enzyme because it will cut at this location. So if you add the restriction enzyme XBA1, it's going to cut at base pair 2506. Okay. Then they will also give you a chart of like this. They'll give you a chart of different restriction enzymes and which buffers work best for them. So you'll select the best buffer to work with the restriction enzymes that you've selected for the experiment. They also will give you a chart somewhere of what the exact cut sites are, what the sequence is that the restriction enzyme uses. Okay, so that's enough for that. Let me go back to my slides. Okay, so if you're not familiar, restriction enzymes are enzymes that act as scissors, and they only cut when they reach their specific sequence. So that list of different restriction enzymes that you saw, they're going to recognize and cut different specific sequences. And just like any enzyme, you need to make sure you give it the correct environment, right? It needs to have the right temperature, it needs to have the right pH, it needs to have, coenzymes, etc., right? Everything it needs to be able to do its job. So you'll select the best buffer for the 2 restriction enzymes that you select. And already went over how to select those with you just now. And so lastly, once you actually perform the experiment, you give the restriction enzymes, you give the correct buffer, and you cut out the GFP gene. Then you will run a gel electrophoresis to isolate that. So you'll have a band that has the plasmid, the remaining plasmid, and then you'll have another band that's just the tiny little GFP gene, okay? And then you'll have to use the gel ladder to know which band is big and which band is small, and you'll choose the small one for your GFP, right? Because GFP is just a gene that's small. The rest of the plasmid is big. Okay, this YouTube video is only 2 minutes long, and it's super helpful for you to see what is a vector and what is a restriction enzyme and how does it work. So everything we've been talking about. So please watch that. I'm not going to show it. Okay, so now that you've isolated RAD52 and you have that in one tube, and you've isolated, purified the GFP gene, and it's in a separate tube, you're ready to put them together to make one unit, okay? And we call that the fragment or gene of interest, okay? So you're ready to ligate together. Ligate is, we use this term because the enzyme is called ligase, and this means to put together, to seal or glue together. It's actually making a covalent bond, and this will be demonstrated in the Labster, as well as that short video clip that I just gave you. Okay, so due to the specific restriction enzymes that we are using, we know that the RAD52 gene is going to come first, and the EGFP gene is going to attach to it after. And we know, if you were able to see the sequence of the RAD52 gene, you would see that only the 3' end, so the end of RAD52 gene, is complementary or compatible with the 5' front of the EGFP. So in other words, the way I've been writing it, with RAD52 coming first and the EGFP coming second, if this was 5' and this was 3', in fact, let me write that on here. Pen, magenta, sure. So this gene, this is the 5' end, and this is the 3' end. So it attached, the GFP will attach to the 3' end of the RAD52 gene in this order, in this way. And that's all due to the restriction enzymes that we used. We were very careful that the that the 3' end of the GFP will not be able to interact with the Rad52 at all. Only this 5' end of the GFP is able to interact with the 3' end of the Rad52. Okay. All right. So next, you've successfully made this fragment, the Rad52 GFP fragment. Now you're ready to insert it into the actual expression vector. Okay. So the expression vector has everything needed so that your fragment, Rad52 GFP, can actually be transcribed and translated in whatever cell you stick it in. Okay. So, and this is also, remember, it's going to remain separate. You're not inserting this into the chromosome of the yeast cell. It will remain separate. Okay. Okay. So what is an expression plasmid? I told you it has all the parts needed to allow expression of your gene of interest. And so that's going to include cloning sites where you can get copies made. Okay. It has a selectable marker, which is typically an antibiotic resistance, so that you can select for the cells that you successfully transformed with the vector. It has an origin of replication so that this can be replicated in the cell. And then it has an operator and a promoter so that you can control the expression of your gene of interest and actually get it made when you want it made. So an operator, it determines whether or not the gene is able to be transcribed, right? And so, of course, we want to make sure we have a really strong promoter so we can easily get transcription of our gene of interest. Okay. So cloning vectors or library vectors, they're not going to have the operator and the promoter, right? Okay. Next. So you've successfully made your expression vector. You inserted the Rad52 GFP fragment into this TRE expression vector. So the TRE expression vector is shown here. This is the vector that we needed to open up and insert and it's here, open it up and insert our fragment, the Rad52 GFP. And so we do it, yay. And I'm not going to go over this slide with you because you can refer back to this if you need to, but I want to stick with our train of thought. So we've successfully made this expression vector and we're ready to get it inside of our yeast cells. So we're going to do transformation to put this TRE Rad52 GFP expression vector inside of the yeast. So we want the yeast to take up this naked DNA, and that's not quite normal, right? So we need to make sure we have competent cells that are willing to do this. You won't have to do that. The simulation will do that for you. And then you get to just zap them. You get to do the electroporation to get the yeast cells to take up the expression vector. And then you're going to let those incubate. And then you have to select for which yeast cells actually took up the plasmid. And you'll use that antibiotic selection process where only the cells that took up the plasmid will have the antibiotic resistance and be able to survive. Sometimes instead of antibiotic selection, we might use like a color difference, right? Anything that will allow you to differentiate between the cells that did and the cells that did not take up the vector. But in this case, it is antibiotic selection. All right, we're almost done. So now you can finally do your test. You've made your expression vector. You got it inside of yeast. You got a whole bunch of yeast that have the expression vector, and now you're ready to see what the RAD52 expression looks like when they're just happy and growing. And then what does the RAD52 expression look like after you hit them with some UV light and damage their DNA? And that's it.